Hello, and welcome to Chapter 13. Today, we're going to talk about the molecular basis of inheritance. We've been dealing with the topics about inheritance for the last few weeks now, but now we're really going to take a much closer look at DNA itself. So, in part one of this lecture, we're going to talk about the basics of chromosomes, DNA, and how DNA makes copies of itself. And as we're going through this, just wanted to point out to you that I will be doing this slightly out of order from the textbook, which is not what I usually do, but I think I like my order a little bit better. So play along with me. If you are reading the textbook, I will be skipping around a bit. So we're going to start out with section 13.3 and talk about how chromosomes are made up of DNA and how they're packed together with proteins to make the structure that we think about as chromosomes. If we think about bacterial chromosomes, they are the same basic structure as eukaryotic chromosomes. It's double-stranded DNA, but the way that the DNA looks, it's actually connected end to end. So it's a circle. It really doesn't actually have an end, unlike eukaryotic chromosomes. We say that eukaryotic chromosomes are linear and bacterial chromosomes are circular. In bacterial chromosomes, that circular DNA is associated with a small amount of protein, and that does help it coil up into this nucleoid region. But with eukaryotic chromosomes, there are a large number of proteins that the chromosomes are associated with. So those are some basic difference between bacterial chromosomes and eukaryotic chromosomes. In bacteria, DNA is supercoiled, meaning it's highly coiled, and it is found in a region in the center of the cell called the nucleoid. And when it does that supercoiling, it in fact is coiling around some of those proteins. So if we take a little bit closer at this coiling concept, the first word that I'm going to introduce to you is chromatin. Now, chromatin is, we say the word, it's a complex. That just means it's a whole bunch of different things together. So it's a complex of DNA and proteins. And in eukaryotic cells, that DNA and protein complex known as chromatin is found in the nucleus. Chromosomes fit into the nucleus through this very elaborate multi-level system of packing, because it turns out that chromosomes, if they're completely unwound, if they're not coiled up, chromosomes are very, very long. And so how do you get this huge molecule to fit into a fairly small nucleus? Well, you've got to pack it. So what we're going to see is chromosomes going through this very, very interesting and complicated kind of cool change in the way that they pack and the amount that they pack during the course of the cell cycle. So the first protein that we're going to really talk about here is the protein called histones. And histones are responsible for the first level of DNA packing in chromatin. There are 4 types of histones are common in chromatin, and that's what we're going to kind of step you through this diagram right here. And we'll look at this diagram a couple of different ways, a couple that I've taken from your textbook, but also we'll be looking at this from some things that I've grabbed from the internet. So the first way that we're going to look at here is here are the little histones. Those are in purple, and there's our DNA in blue. And the DNA wraps around these histones in a very, very distinct manner. And this is called the nucleosome. So a nucleosome is our first level packing, and it wraps twice around 8 histones. And that's our main coiling. Notice that the little tails of the histones are pointing outwards. The next level of coiling happens in the fiber level, and this, a whole bunch of histones kind of wrap and get closely associated with each other. These nucleosomes get wrapped in and are tightly coiled with each other. In the fiber level, they're about 30 nanometers wide. In the histone level, in the nucleosome level, they're about 10 nanometers wide. And of course, our DNA is only about 2 nanometers wide. So we're getting some tightly packing stuff going on here. The next level is the looped domain, and these looped domains they look a little bit like flowers, I suppose. They loop around each other in about 500 nanometer fibers, and that's the diameter of them. And then finally, we'll get them into this chromatid form where they're just really, really highly coiled. And of course, remember that the chromatid form is what we see when our eukaryotic cells go through cell division, mitosis and meiosis. And that happens during prophase, early prophase and late prophase, prometaphase, depending on which version you're looking at. So at interphase, most of the chromatin is compacted into that 38 nanometer fiber that I just showed you. And then, of course, that's going to be continued to be folding in some areas by that looping that I showed you. But even during interphase, the areas of the centromeres, right? That's that pinched in area that we see when chromosomes that X shape, but they have the centromeres at the whole, whether they're replicated or not. Those centromeres, they do in fact stay fairly highly condensed, just like they would be during metaphase. So they kind of stay that way all the time. Condensed chromatin is called heterochromatin. The more dispersed, less compacted chromatin is called euchromatin. And so I've got a couple of visuals for you here. Heterochromatin, that's that more fibery one, and the euchromatin is the more kind of spaghetti or string-like one that I've referred to in the past videos. So when we get this dense packing of heterochromatin, it really makes those areas of the machinery that's responsible for making proteins, transcribing genetic information, pretty inaccessible. We'll learn more about how genes are transcribed in a later chapter. But chromosomes are pretty dynamic. So you can have this one region that's condensed, and that condensed region, it can be loosened, it can be modified, it can be remodeled, depending on the needs of the cell. And histones themselves can undergo these chemical modifications that change the structure of and organization of the chromatin. So it's not like once it's packed that it stays packed. It's a pretty dynamic system. Now, if we go back and think about DNA itself, this is earlier on in the chapter. This is kind of the beginning of the chapter. But if you start thinking about the structure of DNA itself, we can go back to a couple of people who influenced the discovery, who are said to have discovered the structure of DNA. Watson and Crick are the most famous of them because they won the Nobel Prize. But in 1953, Watson and Crick did write a paper introducing this double-stranded, double helix model for the structure of DNA. The full name of DNA is deoxyribonucleic acid, just in case you hadn't heard that before. So DNA contains hereditary information and directs the development of all things in your body: biochemistry, your anatomy, your physiology, and some behavioral traits too. So it's really important that we know the structure of DNA, because that way we can actually learn how hereditary information is passed on during DNA replication. So there's two main things that somebody named Chargaff came up with, and Chargaff contributed to DNA, our understanding of DNA as well. What Chargaff said was that the composition of DNA is with the bases that are present, that the base composition, it varies between species. So no two species have the same number of A's, T's, C's, and G's. But what he did find out that was also very interesting is that in any species, there is a particular percentage of A's and T's and a particular percentage of C's and G's. And the percentage of A's and T's are the same. And the percentage of G's and C's are the same. So the percentage of A's and G's might vary, and probably do vary, but A's and T's were always the same, and C's and G's were always the same. So at this point, nobody really understood why that was true, why Chargaff's rule seemed to be true, until we did discover the double helix nature of DNA. Rosalind Franklin was extremely important in this discovery of the structure because she was the first person to successfully take a picture of the double helix structure. And unfortunately, she did not win the Nobel Prize for her discovery because she actually died from the radiation poisoning that was involved in the photographs that she was taken of the X-ray crystallography. So she died from cancer. But her pictures were seen by Watson and Crick, and they had this aha moment, and they realized that we had this structure that was a double helix. Now, some people say that Rosalind Franklin also realized that it was the double helix, but she just never published, and she didn't get credit for it. So what we've got here is a double helix, and the width of the helix and the spacing of the nitrogenous bases are all basically seen in this picture. The next thing that I'm going to show you is the basics of what they figured out. They visualized all of this just from that picture, which I think is pretty incredible. So a double helix. There's two backbones, and you can see one, I'm kind of tracing it here for you, right here, and the other one goes along this way. Oh, actually, that's the same one I traced before here. We'll do this one. This one right here. So a double helix. And then if you kind of straighten that double helix out, it looks a little bit like a ladder, but it is twisted. And helix just means like coil, like a slinky. A slinky, it looks like a helix. So I suppose if you put two slinkies together, it would be a double helix, right? So I'm going to go through some of the details here with you. For example, the distance from one base right here in the middle to another base right here in the middle is 0.34 nanometers apart. The diameter, that means from the outside of the backbone to the outside of the backbone, that's 2 nanometers. One full turn, if you look all the way around and you follow it all the way to the exact point, mirroring this phosphate and this phosphate, you get 3.4 nanometers. That's 10 bases is in one turn. Other important things to look at is that we have a phosphate group, which is attached to a sugar, which is attached to a base. Now, we have talked about this before when we were talking about the chemistry, biochemistry of living things, and we did go into quite a bit of detail at that time. So I'm just refreshing your memory. If you need more detailed refreshment, please go back and watch that video. That was from chapter 3. So we have this phosphate group, and the phosphate group is attached to the 5' carbon. So here's our carbon right here, and here's the 5' end. Again, if you're having trouble remembering the significance of the numbering system of sugars, you can go back and watch that other video. But there are these 5 carbons in the sugar, 1, 2, 3, 4, 5, and we call them primes. So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. The phosphate is attached to the 5th carbon, or the 5' carbon of the sugar. And the nitrogenous base is attached to the 1' carbon of the sugar. And if we look down here, we can see that the next nucleotide, remember that nucleotides are made up of phosphate, sugar, and base. The next nucleotide's phosphate is attached to the 3' carbon. And these numbers 5' and 3' are going to come up over and over again. So kind of be aware of that. This is one right here, one nucleotide, and this is the second nucleotide, right? And this is the third nucleotide on this side. We say that this end over here is called the 5' end. And it's called the 5' end because the phosphate on this side is attached to the 5' carbon of the sugar, and so that is closest to the end. If we look at this side of the DNA, we notice that this is called the 3' end. And that 3' end is mirrored down here. So it looks like the strand is going 5' to 3' on this side, but 3' to 5' on this side. I can't emphasize to you enough how important this concept is. It's going to be a big part of how DNA replicates that we're going to get into, and so these numbers are extremely important. Let's take a look at why this is the 3' end. And so if we look at the sugar, notice how this sugar looks to be upside down from this one. It is. These two backbones are going in opposite directions, and so they're flipped upside down from each other. On this side, the 3' end is closest to the end, 3' of the sugar. And remember that if we did have another nucleotide right here attached, then it would be attached to this 3' end. But we don't have one there right now. So from this side, we're going from 3' down to 5', and there's that 5' right there. So if we look at the connection between these two sides of the molecule, we see that the nitrogenous bases are attached to each other by hydrogen bonds. Covalent bonds attach the base to the sugar and the sugar to the phosphate, and it attaches one whole nucleotide to another whole nucleotide at the 3' bond right here. And so, but we've got these weaker interactions, these hydrogen bonds holding together the bases. We also, very interestingly, have some van der Waals interactions between the stacked pairs. And it helps hold the molecule together. So we've got some weak interactions holding the bases together. There's 3 hydrogen bonds holding carbon, the cytosine and guanine together, and there's 2 hydrogen bonds holding the adenine and thymine together. But these van der Waals interactions hold between these 2 nitrogenous bases that are not actually attached to each other in any other way except through these van der Waals interactions. So that's the basic structure that Watson and Crick described, and they did it in this level of detail. It was very, very thoroughly described. They even built a model that looks very similar to this. But when we're looking at DNA, we don't always look at it in the same level of detail. Sometimes we look at it in simpler ways, and I'm going to show that to you next. So here's a simplified image. Instead of showing you the details of the sugar and phosphate, it just looks like a ribbon. And there's one side of the ribbon, and here's the other. Notice that the 5' and 3' ends are still labeled. Notice in this one that it is showing you basic shapes of the bases, it's showing you that they fit together, but it's not showing you how many hydrogen bonds there are present. It's just saying hydrogen bonds are holding it together. So the C's and G's kind of seem to fit together. And notice that the C's are a little bit smaller in this structure and the G's are larger. And we see something a little bit similar here in the full side view of the A's being a little longer and the T's being a little shorter. So that's a simplified, it's called the ribbon simplified image. We also have these like ladder looking images. They're flat. They don't show the curvature of the DNA, the helix structure of the DNA. But it does show the phosphate and sugar being attached to each other. Of course, it still shows 5' and 3', 5', 3' on this side, and the other way on the other side. It does show that thymine and adenine are attached to each other, that they fit to each other. It's not showing hydrogen bonds, but it is showing that the adenines are larger and the thymines are smaller. Guanines are larger and cytosines are smaller. So, so that's the flattened ladder one. If we look at this version of the flattened ladder, it just has a ribbon for the backbone, still showing 5' and 3', A's goes with T's, and so forth. We can even have a very, very simplified version of it where you have a ribbon backbone and not even showing you which ones are A's and T's. This is the most simplified version of DNA that you can draw, and that's just two straight lines showing the double-stranded nature of DNA. Two straight lines. That one's very rarely shown. Most of the time we're either looking at one like this, or this one, or this one. If we're not looking at one of these visual images where we're looking at something about the structure, then we're looking at something very, very simple and straightforward. We're just going to look at the DNA sequence. So this one doesn't even show the phosphate and sugar backbone at all. It just, there's a phosphate and sugar backbone. That's pretty much all. Most of the previous versions did show you the sequence of the nitrogenous bases. If we look back, except for these two, you do see the sequence of A's and T's and C's and G's. And of course, we saw that in our first one. This model right here just shows A's, T's, C's, and G's. So this is the DNA sequence version of visualizing DNA. Again, it has a 5' and a 3' end, and they are going in opposite directions, and we'll talk about that more later because it is important for the way that DNA makes copies of itself. So this is the DNA sequence version. Sometimes we only see half of that. Sometimes we'll just see one strand showing you the sequence. But almost every single model that we've looked at so far does show directionality, 5' and 3', and that's because that's so important. So when Watson and Crick and Chargaff and Franklin were all thinking about the structure of DNA, they really didn't understand how the bases were going to be paired up. Like some of their first models had the bases pointing out towards the outside, and some of them they thought that maybe the bases paired with A's always went with A's and T's always went with T's. But that became a problem because it turns out that A's, T's, C's, and G's do not always have the same size. So G's and A's are in the category of purine, and purines are the ones that are the wider ones. They're the ones with a double ring associated with them. And then there's the category called pyrimidines, and that's where the C's and T's belong to. And pyrimidines are made up of only one ring structure, and so if after Rosalind Franklin's picture, we realize that DNA is a uniform width of 2 nanometers, then it's not possible for you to pair 2 purines together and 2 pyrimidines together. And we also know that you can't do like with like, because if these were both A's, right, then there'd be if these two were both A's, then they would be too wide. Just the same way if this was an A and this one was a G, right? Like if this one was a G, too wide. C's and T's too narrow. So it was a mix and match of C's and T's, purines with pyrimidines, G's and A's, purine with pyrimidine. And that was very consistent with that X-ray crystallography photograph that Rosalind Franklin took. Now, I do have a quick mnemonic for you here to remember purines and pyrimidines. So the way I remember it is GA, right? This is the symbol for Georgia. And C and T, that's the symbol for Connecticut, for the state of Connecticut, right? So Georgia is a bigger state than Connecticut, and purines are bigger than pyrimidines. Another way to help you with the same mnemonic is that while Georgia is a bigger state, Georgia is a smaller word than Connecticut, so purine is a smaller word than pyrimidine. If that helps, great. If not, move on. So we're going to talk now about this, how these basic structures are put together. Again, A's going with T's and G's going with C's. And of course, that explains Chargaff's rule of the same amount of A's and T's. But we also notice that that the way that hydrogen bonding happens between A's and T's is very specific. They're held together by these hydrogen bonds, and that's hydrogen and oxygen from these two different molecules, a little bit lower down, nitrogen and hydrogen bonding together. And that makes sense. Both of those are those hydrophilic. They're able to have hydrogen bonds. In the GC structures, there's 3 places where hydrogen bonds can connect them. And so we have 3 hydrogen bonds holding them together. There is one implication of this, and that is that it's actually harder to break apart C's and G's than it is to break apart A's and T's, because 2 bonds versus 3 bonds. It's a little bit different, but that difference can be important when figuring out genetics. So we've got this very clear relationship between the structure of DNA and fitting it together with Chargaff's rules and Rosalind Franklin's picture. One of the really interesting side effects maybe of this structure is that Watson and Crick, when they were writing their paper, they noticed that the specific base pairing that we are seeing, right, between G's and C's and A's and T's, it actually suggested a possible copying mechanism for the genetic material. If you break these apart, right, if you break this strand apart from this strand, then this T can code for an A, right? And this A over here can code for a T. And so we can start making an exact copy by filling it in that way. But, it took them a while to figure all this out. So we'll talk about some possible other hypotheses that were come up with. But I do want to show you the basics of what Watson and Crick really did think was the way that DNA the way that DNA make copies of each other. The word that we use to describe how A's go with T's and C's go with G's is the word complementary. Now, this is not saying, oh, hey, letter T, you look really pretty today. I think I'll come and hang out with you. It just means that they are like complementary angles in math. So A's go with T's and they make a whole line, and C's go with G's and they make a whole line. So this complementary nature allows the DNA to make copies of each other or to reconstruct the other one. In DNA replication, this parent molecule, and that's what we call it, the original one is the parent molecule, it unwinds and it opens apart, and these hydrogen bonds are broken, and each of these 2 sides are still called the parental strand, and they serve as a template for the new complementary strand. The new complementary strand is called the daughter strand, and the old strand is still called the parental strand. So we have a parental strand and a daughter strand together. Notice again that the 5' and 3' stay fairly consistent as we're going along here. Again, we'll learn more details about that in a little while, but just keep paying attention to it. So this thing that I've described to you is the semi-conservative model of DNA replication. In fact, Watson and Crick's model does actually predict the semi-conservative model. Each daughter molecule is half old parental and half daughter molecule, and then we get the newly made molecule. But that wasn't the only idea up there. There were 3 basic ideas, and I'm going to talk to you about each of them and talk to you a little bit about the predictions that would go along with it, and look at a little bit of the science. Your book loves to talk about the science behind these discoveries, and I do too. It's just that we don't have that much time. Just to be very careful with your time. So there was the conservative model, and the conservative model went like this. you've got these two parental strands, right? And they might break apart to serve as a template, but then they reassociate with each other. So the parental strands are always staying together, and they make daughter strands, but the daughter strands stay together. And that happens over and over again. So somehow they would disassociate and reassociate. This was a valid hypothesis that was going around at the time. There was another hypothesis that we call the dispersive model. And what this said is that each strand of both daughter molecules, so we consider about these the daughter molecules, contain a little bit of daughter strand and a little bit of parent strand. So that's the dispersive model. A little bit of dark blue, a little bit of light blue here, same over here. And each time it goes through replication, they would disperse. But of course, we know that this is the way, right? The semi-conservative model, where one parental strand stays in the daughter molecule and the other parental strand stays in the other daughter molecule, and it forms the daughter strand. I hope you're catching the way I'm using parental and daughter differently. This is the parental strand, right? This is the parental molecule as a whole. So this is the parental strand, but this is the daughter strand, but this is the daughter molecule. So yeah, a little bit confusing in language, but I think that it makes sense. As we look at some of the data about how they figured out which one was which, they created these cultures of bacteria, and they gave them these heavy isotopes of nitrogen. We call that nitrogen 15. And nitrogen 15 is heavier than nitrogen 14, the lighter isotope. So what they did was, in the bacteria, they cultured it with heavy nitrogen, and then they transferred the bacteria into a medium with light nitrogen. And they took samples after the first replication, and then they took samples again after a second replication. And when they took samples after the first replication, the DNA was about here, about in the middle of the road in this in the test tube. And then after the second replication from the same bacteria, they had a mix of less dense and more dense. So that's how they eliminated the conservative. Because if it had been conservative, there would have been a mixture after the first replication, and after the second replication, the daughter ones there would have been a lot of those daughter ones and very little of the parental ones. So you would have seen like a difference in the amount of those DNA. And then if it had been the dispersive model, the dispersive model would have ended up having something in the center there to begin with. But in the end, they would have been a mixed bag, and they all would have had some lightness to them. So they would have all been lighter. It's only with the semi-conservative model, that you get both the single band after the first replication and two separate bands after the second replication. If we go back and start looking at DNA replication, which we're going to get into much more detail in the next part of this chapter, we can start to look at bacterial replication. And I'm going to go through this diagram with you a little bit. It's pretty straightforward, but one of the things I want you to know when we start talking about eukaryotic replication especially is that there's more than a dozen enzymes involved in this process, and there's lots of other proteins involved in this process. So that's the first thing. It's very complicated. The next thing that I want to tell you is that even though I'm going to be describing to you the eukaryotic replication in detail, we actually know a lot more about the replication in bacteria because it's so much simpler to study. I mean, let's face it, they only have the one chromosome. So because the prokaryotes only have one chromosome and eukaryotes have many ones, and because prokaryotes have only a few of those proteins binding the DNA, and eukaryotes have many, many proteins because they're circular versus linear. And also, and this is a new thing for you, because there's only one origin of replication in bacteria, and there are many origin of replications in eukaryotic replication. We just know a lot more about the bacterial replication. So let me just go over it with you quickly. And then as we get into the next part, we're going to go into it in more details, sort of looking at it from the point of view of the eukaryotic chromosomes. So here we go. There's one origin of replication, and that's where these little dots are right here. There's just this one area of replication. And I'm going to make this a little bit bigger for you, and hopefully this will work. No, that is off of the screen. I have to make it smaller again. Sorry about that. So notice that our DNA is double-stranded, right? And those two strands pull apart from each other at the origin of replication. As they pull apart from each other, the daughter strands are being built in both directions. So as these strands get pulled farther and farther apart, the daughter strands keep building. Eventually, the two parental strands get pulled completely apart, and by the time that happens, we have the daughter strands completely being made. You can see here the replication fork. That's the location where the two parental strands are being pulled apart. Remember, the diameter is so small, you really can't see the details there as to what is the new daughter strand being built, but you can see the replication fork very easily. And there's a replication fork in the cartoon version. And there it is in the actual photograph version. And we also use the term replication bubble, and that's this region over here that's where the new DNA has been made. As the replication fork moves along the strand, the replication bubble gets bigger and bigger. Well, that's what I have for you for that introduction to chromosomes and DNA and the basics of replication. We're going to take a look at in more detail about how replication works in the next part.